I'd like to acknowledge that the Statewide Biobank is built on the lands of the Gadigal people of the Eora Nation. I'd like to acknowledge the many contributions that Aboriginal and Torres Strait Islander people make to society in Australia, their ongoing role in caring for country, which is extremely important to all of us. And I'd like to acknowledge the leadership of elders, past, present, and emerging. So it's a great pleasure to welcome Dr. Eryn Werry today as our seminar speaker. So Eryn's background is she is a senior lecturer in brain and mind sciences and at the Central Clinical School of the Faculty of Medicine and Health at the University of Sydney. And she's also a research associate in the Drug Discovery Laboratory, which is in the School of Chemistry, Faculty of Science. So Eryn completed her PhD at the University of Sydney, where she studied glial cells, and then she took up a brain sciences postdoctoral fellowship at University of New South Wales studying neurogenesis. And since 2012, she's been researching new therapeutics for brain diseases at the University of Sydney. So Eryn is today going to be talking about organoids, and many of you will be familiar with the fact that organoids are gaining increasing attention, particularly because of their they obviously have many applications in research. They can, they have superiority with respect to other preclinical models, and there's also a lot of interest in terms of their capacity to replace at least the use of some whole animal models as well. So we're really looking forward to Eryn presenting on the New South Wales Organoid Innovation Centre, and we really look forward to learning a lot more about the plans for the centre and some discussion at the end. Thanks again, Eryn.
Great. Thanks, Jennifer, and thanks for the invitation to speak. I'm actually presenting today on behalf of Professor Michael Cassio, who's the academic lead for the New South Wales Organoid Innovation Centre. I've got a few slides from him, which I've notched up on the top left corner for those particular slides. I don't know if it seems like a weird topic to be doing at a Biobank seminar series on organoids, but I think that there's probably good synergy there. So there's been a range of organoid biobanks that have popped up around the world. I'm not sure if there are any in Australia. There's none that I know of, but if I've missed any, please let me know. I'd be interested to find out more about those. But you see them at various research institutes, this one from the Frankfurt Cancer Institute, this one from Cancer Research UK. They tend to be tumour-related organoid biobanks, but we also see them popping up at various different NGOs and also companies. So Merck have a gastrointestinal organoid biobank with 50 intestinal organoids they've developed from both healthy controls and diseased patients as well. So I think it is of interest to the biobank community. Now, just before we get into why people might be wanting to have a biobank related to organoids and what their various uses are, just so that we're all on the one page, I wanted to just quickly go through what an organoid was. So I guess thinking about what it isn't, when we're talking about preclinical models of disease, preclinical studies of disease, particularly at the in vitro level, traditionally that's been done in two-dimensional tissue culture. So that's with a monocellular population, and often people go to great lengths in that kind of research to prove that they've got only one cell type in their tissue culture, whether that's by phenotyping the cells, immunohistochemistry. They're grown in two dimensions, generally on a plastic surface, sometimes coated with various different films or substances. But it's probably no surprise for everybody that that's not how organs actually work. So organs are multicellular. As Jennifer said, I'm from a CNS background, so I've just got a bit of a cartoon of CNS, what's going on in the brain there. You can see multiple different cell types. You've got your neurons, you've got your oligodendrocytes, your microglia, there's astrocytes in there, there's even blood vessel related cells, all those cells together. They're growing in 3 dimensions, and they're not contacting plastic unless you've had an unfortunate interaction with a plastic object. They're generally contacting other cells, and that's a big input into their function, release of other molecules from other cells and contact molecules as well. And they also have complex cytoarchitecture within the organs. They grow in various patterns, various layers, which are all important to the function of the organ. So the traditional 2D tissue culture model isn't great at predicting and replicating what's going on in an organ. And so organoids, I guess, are a little bit of a midway point. So they tend, or that they're multicellular. So you can see here we have a mock-up of, or we have a staining of a cerebral organoid. And you can see from the various different colors of the cell markers that there's multiple cell types. And if you have a look on the left here, we've got a Petri dish, which has a cerebral organoid that's been growing for 3 months there. And they've been analysed for the various different types of cells that you find in there. And you can see that there's so many different types of cells that grow in that organoid, both immature and mature neurons, precursors, inhibitory neurons, projection neurons, glial cells. So they're definitely multicellular. They're growing in 3 dimensions. All the cells are contacting each other. And you can see from this picture up the top here that they have a self-organized cytoarchitecture. So you've got some rosette-looking structures up the top, and then a different cell type all distributed down the bottom. That the cells aren't randomly growing. There's a number of different sources that can make these organoids. So I mentioned the cancer biobanks before, the cancer organoid biobanks. So you can take a biopsy from a cancer and grow that into an organoid, which is just on the right of this picture here. Various different organs have adult stem cell populations in them. And so if you take a biopsy of that tissue with those adult stem cell populations in them, they can then grow into organoids. Embryonic stem cells are another source. So embryonic stem cells can make any cell in the body. And so with various different mediators that you can put on them, you can drive them to become organoids representing different organs. But probably the most popular way to do this is with induced pluripotent stem cells. And for those of you who are unfamiliar with that term, it was once thought that our stem cells in our body, they decided what they wanted to do when they grew up. They decided to become, for example, skin fibroblasts. And that was a commitment that they couldn't go back on. But what Shinya Yamanaka found about 10 years ago that won him the Nobel Prize was that if you take some of those terminally differentiated cells, in particular, for example, skin fibroblasts, and you put in 4 different genes that are found highly expressed in stem cells, you can drive these cells back into a stem cell-like state. They're pluripotent, so they can make almost any cell in the body, and they can replicate theoretically forever. And so one advantage of using iPSCs is you can take skin cells from somebody who doesn't have a disease and skin cells from somebody who does, and then derive pluripotent stem cells from those, and then make them into any particular cell type of the body you want and compare disease state with the non-disease state. So you can make it into the different germ layers, and then with various different chemicals, all these different types of organoids have been made from iPSCs. Okay, so I've spoken a little bit about the anatomical differences between organoids and other models. But what about the functional differences? Do cells in the organoids act any differently to what they might in a 2D culture or an animal model? I'm from a CNS background, so I'm going to give you 4 examples from a CNS context. I just don't trust myself to talk with any great confidence about other organs. So the first one I'm going to give you is a cancer-based example, so glioblastoma. So glioblastomas have quite a strong adult stem cell population. Glioblastomas are quite a high-grade cancer, quite invasive, quite difficult to treat. And part of that is because they have quite a strong stem cell population. OCT4 is a marker of that. And you can see in a 3D organoid from glioblastoma cells, there's a much higher expression of that adult stem cell population marker than there is compared to when they're grown in 2 dimensions. Glioblastoma is usually treated 3 ways. So the first is, if possible, surgical resection, also radiation and chemotherapy. And the particular chemotherapy drug that's often used is temozolomide. But the problem with that is that cells develop resistance quite quickly. What we've got here is a measure of potency for temozolomide to be killing these cells. And we have cells grown in 2D in the light colored bars, cells grown in 3D in the darker bars. Pharmacology is a bit backwards. So a high potency means that, like a high number of potency means that it's not very potent. And so you can see in every case, temozolomide had a worse potency in 3 dimensions in organoids compared to 2D. So these organoids of these glioblastoma cells seem to reflect a greater development of temozolomide resistance, which you would expect when you look at the intact cancers. Okay, so that's a cancer example. Onto a viral example. So up the top right, we're coming up to the Paris Olympics, but if we go backwards a few Olympics ago to the Rio Olympics, you might remember seeing these kind of pictures because there was a Zika virus outbreak around about that time, and they were trying to get rid of all the mosquitoes because it's a mosquito-borne disease. One of the things that Zika does is cause microencephaly. So that's a reduction in the size of the growing brain if it infects fetuses that are growing. And you can see that here with these children. So their heads develop a smaller size. And the impact of Zika virus on brain growth was first really seen when it came to organoids. So they grew organoids, infected them with the Zika virus, and found that if they're infected with Zika, they grow much smaller compared to if they weren't. And obviously that can't really be seen in 2D because they don't grow in 3 dimensions. Another thing about the Zika virus, often when humans are infected by viruses, you get a strong interferon type 1 response, but the Zika virus inhibits that. And that is modeled best in organoids compared to in 2D. So you get quite a strong interferon response on Zika infection when cells are grown in 2D and a very small response. It's still statistically significant, but much smaller than when those cells are grown in 2D. And if you take the organoid cells and you dissociate them out of 3D and grow them in 2D for a while, you actually get an even stronger interferon response. So it seems that being in 3D is necessary for this model to be able to recapitulate that Zika attenuated interferon 1 type response. Our third example is from neurodegeneration, so Alzheimer's disease. Many of you probably know there's 2 key pathological hallmarks in Alzheimer's disease. So you get too much amyloid beta 42 being released extracellularly. And if that concentration builds up too much, then they form these aggregates that show up as amyloid beta deposition in postmortem tissue and also in people with the disease. You can pick that up with PET scans. And then also in our neuronal cytoskeletons, tau is a protein that helps binding and stabilizing the cytoskeleton there. And in Alzheimer's disease, tau gets hyperphosphorylated, which causes it to detach from those microtubules, and that causes the microtubules to break down, and you get neurofibrillary tangles, which is the other main pathology that you see. Traditionally, this isn't modeled very well in animal models. So the amyloid depositions seem to be modeled quite well, but tau pathology, those neurofibrillary tangles, don't often show up in animal models of Alzheimer's disease. So this is what happens when you make iPSCs from Alzheimer's disease donors. You let them grow for 60 days. You see robust amyloid beta deposition, and also that phospho-tau pathology shows up. So it seems like these organoids are better able to recapitulate some of the key hallmarks in Alzheimer's disease compared to many animal models. Some of you also may be very well aware that there has been a real struggle with finding Alzheimer's disease drugs that work in the clinic. So many drugs over the last 20 years have cured 2D tissue cultures of Alzheimer's disease. They've cured animal models of Alzheimer's disease, and then they've then gone into the clinic and failed. And BACE1 inhibitors are an example of that. So BACE1 inhibitors work on one of the enzymes involved in liberating the amyloid beta 42. And so the thought was that you could inhibit those enzymes and have less being secreted and therefore less building up. BACE1 inhibitors, again, they cured animals of Alzheimer's disease, but when they went into the clinic, so far they've all failed. This is an example of what happens when you treat one of these organoids, the Alzheimer's organoids, with a clinically failing BACE1 inhibitor. When you treat them for 30 days, you get no impact whatsoever on the neurofibrillary tangle pathology and the phospho-tau immunoreactivity. If you keep treating for 60 days, you start seeing an effect, but it's quite subtle compared to the effect that you see in the other models. So it's looking like these organoids have quite good predictive validity compared to some of the other preclinical models. So we've had a viral example, a cancer example, a neurodegeneration example, and I'm going to finish these examples with a genetic disorder. So it's very rare, but a number of families around the world have a knockout of pitram-1. So that's petrolism metallopeptidase 1. And that plays a key role in getting rid of mitochondrial targeting sequences from proteins. And so with this deletion, it means that there's an accumulation of proteins outside the mitochondria where they aren't necessarily found there a lot. One of the things that builds up because of that is amyloid beta. And so these families that have this pitram-1 genetic deletion show cognitive decline with age and build up of amyloid beta in their brain. So when iPSCs taken from the people that have this disorder are grown into neurons in 2 dimensions, you can see quite nice neuronal cultures there. BTA1 is a stain for that amyloid beta accumulation. And you can see that there's no amyloid beta accumulation when they're grown in 2 dimensions. But when they're grown into 3D organoids, you don't see any in the wild type. But when there's a deletion of that pitram-M1, you can see quite robust amyloid beta forming. And when you quantify that, there's quite a large difference there. So it seems like the pathology only shows up in this genetic disorder in the 3D organoids and not in 2D. Okay, so in an effort to give you a balanced picture of organoids, no model is perfect, and there are some limitations with organoids. So compared to, for example, 2-dimensional culture, it takes a lot longer to get these to a stage where you can test them, and it's more expensive. You need the culture media and supplements for much longer. Although I would probably argue that in some circumstances, this is probably cheaper than the animal models. Another limitation is with vascularization. So often these organoids don't contain blood vessels, and that can be a limit for a couple of reasons. Firstly, because things like endothelial cells and parasites release important mediators that in many instances do contribute to disease. So without those vessels in there, it's an incomplete picture of a particular organ's function. And also the 3D structures, they get fed by the medium that they sit in, and that medium can't always penetrate to the middle of that organoid. And so sometimes you see a necrotic core forming in the middle of the organoid. The cells start dying because they can't access medium. So vascularization would be really handy to be able to enable all of that organoid to get food. So people are trying to find a way around that. This is a particular study where people grew organoids from a human iPSCs into a cerebral organoid. And then they implanted that into a rat brain. So you've got the brain here. The organoid, the human cerebral organoid had been transfected with GFP. So you can see it sitting there. And over the course of 10 days, it grew neurons out that started synapsing with neurons in that rat brain. And I'm not sure how clear this will be on the slide, but again, over the course of 10, 20 days, you start seeing blood vessels, so here, and here, starting to grow into that organoid and innervate it with blood. Another limitation with organoids is reproducibility. So again, people are working on ways to try to make this less of an issue, but because the organoids are self-forming, they might differ. If you take a bunch of iPSCs and split them up into different wells and grow different organoids, or grow organoids under the same condition from the same starting cells, each organoid will be slightly different. They'll contain different proportions of cell types. The cytoarchitecture might slightly differ. The overall shape might differ. And so that becomes tricky when you're doing something like drug discovery and treating each different well with a different drug. You can't be 100% sure that any differences you see are because of the drug as opposed to slight differences in the organoids. So another approach that can help with that and still allow cells to grow in 3D is 3D printing. So what I'm showing you there, that picture on the bottom left, is a cell printer from Inventia Life Sciences. It's nice that it's bucked the trend of the grey box, the beautiful purple. And some work that I'll show you with a student of mine, a PhD student who graduated last year, Dr. Michael Sullivan. He experimented with different inks, bio-inks. So these are hydrogel bio-inks containing different cell adhesion peptides, and he mixed some iPSC-derived astrocytes with that ink and printed them into little plugs. And you can print 96 different wells, and then they all contain the same amount of cells. You can print different types of cells all together, so you can get that multicellular configuration, and then you can control the proportions and make them even across wells. So that's handy for something like drug discovery. And he found that when he did that, he also did this with iPSC-derived neurons. And when those neurons were grown in 3D, you had a much greater proportion of signaling events than you did when they were grown in 2D. So they seem like they're more functionally active in 3D. So I guess the 3D printing also has the downside. This bio-ink isn't necessarily exactly what happens in the body that you might see in an organoid. So all these 3D models have their pros and cons, and perhaps in the future, we might be moving to a situation where you do your particular preclinical 3D model in various different ways. So you might have an organoid testing, and then you might have a 3D printing testing, and that will provide converging lines of evidence for your research question. Okay, so how are people using organoids? There's some really interesting, fascinating ways. This is a couple of examples of those. Some of you might have seen this in the news a few weeks ago. A lab in Japan created this robot, and the robot was able to almost be self-autonomous. It could react to light, it could walk around obstacles, and when they flipped the hood, there was an organoid in there. This is not the actual organoid they used. It's sort of a recapitulation of what they think it would look like, but it was that organoid that was providing that computing power to allow it to react to external stimuli. The Daily Mail jumped on that and put out quite an inflammatory headline. But there's a company down in Melbourne, Cortical Labs. Some of you may have seen this on the news as well last year. They used iPSC-derived neurons just in 2 dimensions, and they showed that they were able to teach those neurons how to play Pong by themselves, the computer game Pong. So those neurons were able to defeat Pong. And so the idea that they're working towards is, rather than silicon computer chips, having computer chips with actual physical neural networks from these iPSC-derived neurons, and then moving towards having organoids potentially eventually. So that's, I guess, a biocomputing use. People are also using it to study neurodevelopment. So I'm going to show you some studies by Alison Muotori now. He grew cerebral organoids for 10 months. Anybody that's done any tissue culture hopefully will find that very impressive, to be able to keep something in an incubator continuously for 10 months without it getting contaminated. He picked 10 months to roughly mirror human gestation period. And you can see that over the course of that time, the mean firing rate of neurons in that cerebral organoid increased a lot compared to 2D, where there was hardly any increase up to 20 weeks. He then quantified and classified the different neuronal firing patterns. He found as time went on, the firing became more complex. And then he analysed the complexity of the different firing patterns within the organoid at 10 months and compared that to EEGs of preterm neonates. And he found that there was quite a lot of overlap in the different aspects of the signalling. So it looks very different to an adult EEG, not as complex, but the organoid signalling has similar type of pattern to the preterm. And so he said that this provided quite good evidence that over that 10 months, we can recapitulate some similar processes to what happens in human neurodevelopment. And so people have then gone on and made organoids from autism spectrum disorder donors, other neurodevelopmental donors, and use that to explore what might be happening as the brain develops to understand the disease process better. Alison Muratori has also done some interesting work understanding physiology, perhaps subjecting these type of models to stimuli that you might not necessarily be able to subject other models to. So he's interested in what happens to the brain as it goes into space. And of course, astronauts go into space and you can do EEGs and fMRIs on their brain as they come back, but that doesn't tell you what's happening at the cellular level. So he's worked with SpaceX to send some of his organoids off into space to understand what happens at the cellular level. And they haven't published these findings yet, but initial reports are that there are quite big changes that happen to these cells as they go into space. Personalized medicine is another big application of these organoids. So I'll give you two cancer examples now. The first was with pancreatic cancer. So this cancer discovery paper generated organoids from 78 pancreatic cancer patients. So these were from biopsies of the pancreatic cancer. And then the patients and their organoids underwent treatment with the chemotherapy drug gemcitabine. So that's what the GEM stands for here. And they stratified the patients based on whether or not their organoids were sensitive to gemcitabine. So in blue, we have people whose organoids were sensitive, that gemcitabine killed off the cancers in the organoids. And in red, the organoids didn't react. And you can see that the survival rates of the patients with gemcitabine sensitive organs were higher than those with non-sensitive organoids. So this could be a potential way of screening people to know whether or not particular chemotherapy drugs will have an effect. But also in doing this, they were able to find some genetic signatures which predicted which patients were sensitive and which patients weren't sensitive. And so that could also be a useful screening in the future. I guess a trend in cancer treatment at the moment is immunotherapy. There are a few different types, but one of them is T-cell transfer therapy. So getting T-cells, growing them up, T-cells that might attack the cancer, and then putting them back into the body. What they found here was when they made organoids from this particular intestinal cancer patient, so they did this with a number of different patients, they grew their T-cells up either without the presence of these organoids or in the presence of these organoids. And they were able to find a lot more higher proportion of T-cell active cells in the blood that had been grown with the organoids. And those T-cells were able to more effectively kill off those cancers. So that could be a way of boosting T-cell transfer therapy. And then the last application I'll go through is drug discovery. So I guess I'll start with a pretty obvious point. Drug discovery has some really great things about it. It's made some really great drugs over the years. So these are the top 5 prescribed drugs in Australia between July 2020 and June 2021. So we have some statins, some reflux drugs, and a sort of hypertension, heart failure drug here. If we add up all the scripts for these, that was 50 million scripts over the course of a year just for those 5 drugs alone. So this process does have some big impact on people, but it also has some downsides as well. And that's particularly with its inefficiency in coming up with new drugs. So it's potentially around 10 to 12 years by the time you find a structure for the first time to get it through all the different phases, preclinical testing, clinical testing, and then through the regulatory process. It can take about 10 to 12 years, costs a lot of money. Predictions vary, but this particular paper said $1.3 billion to get one drug approved by the FDA. So each of these ones went through this process. And there's also a lot of redundancy. You start off with a lot of different drugs, and then they keep failing at each step before you end up with one that actually works. What we ideally need is better initial preclinical testing methods to be able to make better decisions about what compounds to move forward. And if we look at an analysis of why drugs fail when they hit the clinic, particularly this study looked at phase 3 trial fails, efficacy was the biggest reason. They found 55% of clinical failures of drugs are from efficacy problems. And that suggests that the tests that we're using preclinically to predict which drugs are going to work in the clinic need to be improved. And so it's hoped that maybe organoids could do that. And so again, what we would be doing would be getting iPSCs from patients, iPSCs from healthy controls, making them into whatever organ that you're interested in terms of drug development, and screening those drugs on the healthy controls versus diseased. And this is a Nature Com paper that took that model with Alzheimer's disease. So they grew up over 1,300 different organoids. They grew them to day 60, and they checked that they had those hallmark Alzheimer's disease pathologies, amyloid beta, tau deposition. And then they tested a whole bunch of already FDA-approved drugs, so drug repurposing. And they were able to find 3 or 4 drugs that managed to reverse these pathologies of Alzheimer's. And the advantage of repurposing is that they then don't have to go through all of that FDA approval process. To be able to do that, though, to generate 1,300 organoids requires a lot of capacity. That's where the New South Wales Organoid Innovation Centre comes in. So the New South Wales government's Emerging Industry Infrastructure Fund recently granted the New South Wales Organoid Innovation Centre $2.5 million to get established. So as I mentioned at the start, the academic lead of this centre is Professor Michael Cassio. So he's based at University of Sydney. The Organoid Innovation Centre is a collaboration between USyd, UNSW and CMRI, with Shafan Waters heading up the UNSW node and Anai Gonzalez Cordero heading up the CMRI node. And so the UNSW and CMRI nodes will have leadership in producing the organoids, and then the USyd node will house the infrastructure, and that includes the Cell Express AI. It's a molecular devices system which allows automated culturing of these organoids. So everything from the incubation, the plating of the cells, the feeding, checking that they're looking okay and growing in a way you'd expect, and doing measurements that you might need for your experiments, and passaging. So anyone that's been involved in this work before, these cells are quite prima donna-y. They like feeding 7 days a week, unlike the traditional 2D culture where it's often Monday, Wednesday, Friday. So being able to get your weekends back alone is very exciting with this machine coming in. But there's also benefits with that automated process of increased reproducibility. So I'll just show you a quick video that goes into this system a little bit more.
Cell culture has been at the core of drug discovery for nearly a century. However, clinical trials are costly and frequently fail because of inadequate models that don't accurately represent humans. Drug screening and toxicity results using 3D models are more physiologically relevant and therefore more accurately predict clinical trial outcomes. These cell culture protocols are much more complex, time-consuming and error-prone. Mistakes can lead to weeks of time lost and the waste of expensive reagents. Introducing the CellExpress.ai automated cell culture system, a revolutionary cell and organoid culture platform that automates the holistic process of cell culture and differentiation for imaging and biochemical assays. It makes the process of cell culture a reliable, quantifiable and predictable science, utilizing AI decision-making and high-content imaging in a fully automated environment with 24/7 hands-free operation. CellExpress.ai creates an ecosystem where more complex, biologically relevant model systems can flourish. It ensures reproducibility for standard and complex cell-based assays. Automated feeding and passaging reduces human error, removes variability, and increases confidence of success. Reclaim scientists' time from meticulously monitoring their assays to analyzing quantifiable results. CellExpress.ai runs autonomously, provides real-time feedback, and alerts users to milestones. Welcome true productivity with the ability to create reliable 2D and 3D cell cultures at scale. With cell culture taken care of, you and your highly trained staff can focus on better understanding disease and developing groundbreaking therapies. Discover the future of cell culture and unlock the full potential of your research with CellExpress.ai.
So that's going to be a core part of the New South Wales Organoid Innovation Centre. And there's also some high content imaging systems that will go along with that, some plate readers. And the node will also house that Inventia cell printer as well. So Sydney Analytical will house this. Yeah, that's about all I had to say.